Hello, my name is Kaleigh Roe, and I'm going to be presenting my research on bioengineering cassava for increased vitamin A expression through Agrobacterium mediated transformation. Cassava is a very important crop in developing countries and is readily available and consumed. However, cassava is lacking nutritional value. The Arabidopsis AtOR gene is reported to be involved in carotenoid biosynthesis and is known for its ability to express and increase vitamin A levels. Carotenoids are metabolites that contribute to the yellow, red, and orange pigments that crops express. Carotenoids have physiological functions which aid in preventing photooxidative damage. Arabidopsis orange proteins are considered a major posttranscriptional regulator of the phytoene synthase complex, which is PSY. This plays a role in controlling carotenoid biosynthesis. The purpose of this research is to increase the nutrient levels, specifically vitamin A, in cassava by expressing AtOR and AtPSY individually as well as together in the same transfer DNA. 3 constructs will be produced, a construct containing AtOR, PSY, and a double construct co-expressing both AtOR and PSY. During the first attempts, root-specific promoters GBSS and Patatin I were used. Currently, we are working on generating constructs with expression cassettes of both PSY and AtOR with a new constitutively active 35S promoter. The single construct of PSY AtOR has been moved forward in the process of undergoing Agrobacterium mediated transformation as well as FAC calli regeneration. Cassava is a very important starchy and tuberous crop in the developing countries of Africa, Asia, and Latin America. However, cassava is poor in proteins as well as vitamins. Cassava is a rooted vegetable and is known for its characteristic of a widely understood genome. AtOR is an orange protein classified as a carotenoid and is known for the ability to express and increase vitamin A efficiency. Orange proteins are regulators to the carotenogenesis and are pigments in plants but act as antioxidants in humans. Carotenoids are metabolites that contribute to the pigmented colors that plants express. Carotenoids have physiological functions which aid in preventing photooxidative damage as well as increasing vitamin levels in humans. Arabidopsis orange proteins are a posttranscriptional regulator of the phytoene synthase complex. This plays a role in controlling carotenoid biosynthesis. PSY is a phytoene synthase and is a rate-limiting step of the carotenoid biosynthetic pathway. 3 constructs were assembled, one expressing just AtOR, one expressing just AtPSY, and a double construct co-expressing AtOR and AtPSY in hopes to further enhance carotenogenesis. We will focus here on AtOR, which was paired with a root-specific promoter, GBSS. Having a root-specific promoter is essential for ensuring optimization of the genes. Once a construct is assembled and positive, it is transformed into cassava where embryonic cells will differentiate to express AtOR. Once transformed into cassava, we expect an increased amount of expression of vitamin A, making cassava a healthier and more sustainable crop. Cassava is currently a staple crop in Africa and South America, as noted before, and it is a major part of the diets. However, it lacks the nutrients needed to classify as a sustainable dietary nutrient source. But by increasing the rate of carotenoid biosynthesis and vitamin A content through AtOR expression, this should yield increased production of antioxidant and vitamin quantity within the plant to incredibly benefit the area and the individuals who utilize cassava. Experimental methods procedures are as following, starting with the generation of the expression cassettes listed previously. Each expression cassette was assembled via digestion with specific restriction enzymes. The gene and promoter were digested from PUC57 and then ligated into PSAT modular vectors. Once positive in our modular PSAT vector, the promoter and gene were digested again using restriction digest and ligated into our final binary PSAT vector. Once that was positive, we moved forward to our production of friable embryos. Cassava was maintained and controlled in a 28°C growth chamber. Stems with at least one shoot were extracted with a blade and then cultured onto MS media. Friable embryogenic calli were generated from cassava immature leaves. The binary vector containing the expression cassette, which is our PSY GBSS AtOR cassette, was introduced into Agrobacterium LBA 4404A strand for FEC infection. Transformed cells were selected on a media containing paramycin. Paramycin-resistant calli were cultured on a germination media to generate putative transgenic lines. So far, the single AtOR has been used to transform cassava, and putative transgenic lines are being germinating for maturing embryos. Our results show that the germination of putatively transgenic lines from paramycin-resistant calli were observed low frequency of embryo maturation and regeneration, which could be due to conditions used in embryo maturation and germination. However, we expect to obtain sufficient lines for characterization. Our future directions include that Agrobacterium mediated transformation will be done and the T0 lines of putatively transgenic cassava will be grown. Once these putatively transgenic lines have been regenerated, the plants will be transferred to soil and genomic DNA will be extracted. PCR will then be used to validate the insertion of AtOR, and GPCR will be used for evaluating the gene expression. A protein assay will follow to be used to measure the vitamin A content expression. Thank you for listening.